miRDP2 can be used to accurately identify plant microRNAs that are important transcriptional regulators in plant development and are crucial for responding to environmental challenges.
miRDP2 requires a markedly shortened running time and exhibits an excellent performance in both sensitivity and accuracy, especially for predicting microRNA in plants with a large genome.
False positives and long processing time are challenges in plant microRNA annotation. By adding a new filtering strategy, overhauling the scoring algorithm, and integrating stringent criteria, miRDP2 can overcome these issues.
High-confidence microRNA annotation is fundamental for discovering the role of microRNA in regulating diverse functions of the genome. Step-by-step guidance can be helpful for first-time microRNA annotators. For experienced researchers, the method is useful for understanding the advantages and benefits of using miRDP2 over other tools.
To install the miRDeep P2 package, navigate to the miRDeep P2 webpage and fetch the tarball files. Then extract all contents of the downloaded file into one folder and set the folder path to path. To test the miRDP2 pipeline, download the test data and the expected output containing one formatted GSM sequencing file and one Arabidopsis thaliana genome file, and move all of the downloaded files to the current working directory. After extracting the compressed tarball files, build the Arabidopsis genome reference index and